Well, good afternoon, everyone. I think this is the last session, last panel of the day. Welcome. This is the Gene Therapy and Modified Cell Therapies panel. It's somewhat of a new area for this stem cell meeting on the Mesa. It's somewhat of a new area for ARM, and I hope that the panel that we have for you today is going to talk a little bit about why we're enthusiastic about the field of gene therapy and modified cell therapies. Before we begin, just for brief introductions, my name is Edward Lanphier. I'm the president and CEO of Sangamo BioSciences, and I'm the moderator of the panel today.
Faraz Ali, VP of Commercial Development and External Affairs at
Bluebird
Bio.
Do that again. Your mic wasn't on.
Faraz Ali, VP of Commercial Development and External Affairs at Bluebird Bio.
Hans Hull, SVP of Business Operations at Avalanche Biotechnologies.
Jeff Coder, Head of Business Development at Voyager Therapeutics.
I'm Doug Kerr. I'm a neurologist, neuroscientist, previously in clinical development, now in corporate strategy at Biogen IDEC.
We almost sat randomly in the order on the slide.
I know.
Completely remarkable.
We can still pull that off.
Well, we could, they might get up and dance. Who knows? All right, so we'd like to keep this as interactive as possible. We intend to keep it interactive up here. Not surprising to any of you, we had a good call ahead of this, and so have some topics that we're planning to discuss. But at various moments in this, I'd like to stop and make sure that we create opportunities for questions and conversation with you all and make it as productive for you as we can. So the first two general topics we want to talk about is kind of the oversight or overview of in vivo gene therapy, and then followed on that the area of modified cells or ex vivo gene therapy. So to kick it off, I just thought, maybe Jeff, you could start with, and given perspective from Voyager, maybe you can even start there if you want. But can you just a kind of an overview of opportunities, but challenges, difficulties, issues that we all face in vivo gene therapy?
Yeah, sure. At Voyager, obviously, we're focused on AAV gene therapy for CNS disorders, and our feeling is that AAV has really risen to the top of the heap as the vector of choice for in vivo gene therapy for a number of reasons. Don't need to necessarily go into on this panel. And as you think about the opportunities and the challenges as it relates to in vivo gene therapy, I think oftentimes the opportunity is skipped over and we rush right into the challenges. I think the opportunity is a fantastic one, which is we have a real opportunity to have a meaningful impact on the lives of patients. Some of the early data, Bluebird's data in beta thal, some of the ophthalmology data, hemophilia data, et cetera, really suggests that this type of therapeutic modality could have an impact on patients where they're very severe disorders and where there was little or limited therapeutic options in the past.
Yep.
And it sounds a little bit trite, but I think it's important to put that front and center. Now, the topic that we all love to kind of armchair quarterback, and that's the challenges.
Sure.
And there are certainly a number of them. Scalability, to kind of hit on that one first, is something we talked about on the conference call. And here it's about manufacturing high-quality recombinant virus in a scalable way. And depending on the therapeutic target that you're going after, the indication you're going after, that's going to have a direct impact on the amount of virus you need. Generally, how we think about it for some of the ophthalmology indications, relatively small amount of virus is required. As you start to think about CNS indications, we have indications where we're going direct injection into the brain parenchyma. You need incrementally more. When you start talking about intrathecal dosing, direct to the CSF, you're taking another step above that. And then when you start talking about systemic administration for a variety of disorders, you're talking about a whole lot of virus. So at Voyager, we're focused on a baculovirus-based production system. We actually recruited the inventor of the system from the NIH, a guy by the name of Rob Cotton. Great guy, he's done great work, and he's focused on implementing that, both from a research grade, but more importantly, a GMP perspective. There's three other challenges that I think all are more interrelated, and as we talked about them, immunogenicity, persistence of gene expression, as well as durability of clinical benefit. I think as we think about immunogenicity, we tend to bucket it in one of two categories. There's immunogenicity against potentially the transgene that you're delivering. There's also immunogenicity, more obvious, against the vector itself. And if you're talking about truly kind of one-time therapy, becomes more of an issue if there's pre-existing immunity. Obviously, if you had neutralizing antibodies against your AAV serotype of choice, you're going to want to know that before you go in from a therapeutic perspective. If you're in a situation where that may necessitate redosing, I think there, obviously, the generation of neutralizing antibodies that I think has been pretty convincingly shown is going to be a little bit of a challenge. So can you identify capsids or vectors that have the same tropism that you're going after, but maybe a little bit of a different immunogenicity profile? And there, it's a little bit of an open question. Are the neutralizing antibodies cross-reactive across the capsids, et cetera? But definitely an area of focus for Voyager, and I know a number of the AAV gene therapy companies. That feeds right into persistence of gene expression. And again, how long will you have to go before you need to redose? One of the reasons Voyager was so interested and focused on CNS is because we are targeting terminally differentiated neurons. So even though AAV doesn't readily integrate into the target cell genome, we've shown through collaborators that you can get persistent gene expression, non-human primates out beyond 10 years, and previously completed clinical trials out beyond 5 years. So not forever yet, but it's certainly indicative of a long-term gene expression. And finally, as we talk about durability of clinical benefit, a little bit different in terms of the way we think about it. You can think about it as persistence of gene expression, but the way I think about durability of clinical benefit is kind of getting over that threshold of gene expression to have a therapeutic impact. And I think here you think of the hemophilia studies that have been done, where you actually can get a relatively small percentage of endogenous gene expression via the transgene factor and have a therapeutic impact. Voyager's going after a number of autosomal recessive monogenic disorders where the genetics are very instructive. If you have one healthy copy of the gene, that's enough to offset any sort of phenotype. So as we think about going in with in vivo gene delivery, if we can kind of get to that threshold, we feel reasonably confident that we'll have a therapeutic effect.
Good. I mean, Hans, so, many of those are a good list of some of the issues, challenges. Maybe we can review those in a minute, but some of those aren't as big a factor in the eye. Is that fair?
So yeah, I mean, as Jeff pointed out, I think that's true on the scalability side. Although I think overall, just to sort of touch on scalability, I think that's a good news area where there's good progress in many systems. We've also seen really positive results with the baculovirus system. Right. If you sat in on the AGTC presentation, I think they seemed pleased about their recombinant herpes approach. So, when you think about the first generation of AAV companies, a lot of them really, struggled with this issue and getting the amount of investment right. And I think that's an area where the field has really matured. I think with respect to duration of clinical benefit, at least the clinical data in the eye is encouraging. If you look at the data out of Penn with the Lieber study, I think we're out to 6 or 7 years in the first eye on that study, and still seeing, good clinical benefit. I don't think they're doing vitreous taps to see if there's expression, but you'd expect to see that. And then the dogs are out to 11 years. So that may have to do with the immune privilege in the eye and sort of the different nature of that compartment. But I think that is something where we feel pretty good about it.
Yeah, and I think in CNS and in the eye, depending on turnover, persistence it's a reasonable strategy. I mean, in terms of long-term effects.
And as you get to these other tissues, mitotic tissues out in the periphery where an immune response is likely to be more exuberant, these challenges are magnified. So people have done, a really smart thing in picking those disorders in which you may not have as many challenges in certain of these areas that you talked about. And then as we get into second and third generation, hopefully we'll know more, whether it's directed evolution for biodistribution, whether it's cloaking strategies to avoid immunogenicity. I think we'll have a better sense for whether we can redose. We're not there yet, and so kind of the lower hanging fruit, which is still pretty high fruit to pick, is to go where these guys have been going.
I'm going to quote you on that one. Good. I want to pause in a moment and come back and see if that discussion was sufficiently robust in terms of AAV. We haven't discussed other in vivo approaches, but let me turn to the other side and then compare and contrast a little bit. And so Faraz, can maybe do a similar kind of pros and cons, opportunities, challenges on the modified cell therapy side of things, ex vivo?
Yeah, sure. So I think the first thing to do is to make sure that we're categorizing the world properly. Most people sort of typically associate in vivo AAV only and ex vivo lentiviral vector. And as I think we've looked at the presentations that preceded the session, the world's a little bit more complicated. There are some lentiviral in vivo, and that's what some of our colleagues who are working on the eye are doing. And then there, ex vivo, it doesn't automatically mean lentiviral vector. So if we look at the world of ex vivo, what are we typically talking about? We're talking about taking the patient's own cells, doing something outside the body, and then putting those cells back in. Those cells have historically tended to be either hematopoietic stem cells or T cells. So the T cells in the case of Carl June and other groups working in CAR T cell technology, and for hematological malignancies, CLL, ALL, there's been some great data in that. In the case of hematopoietic stem cells, our program in beta thalassemia, our program in HIV, and our program in sickle cell disease, adrenal leukodystrophy, other people's programs in Wiskott-Aldrich. So there's a range of indications which are taking this ex vivo approach, manipulating the hematopoietic stem cells. And so, one point we all need to understand is we got to get the right tool for the right job. So in ways, we're not competing with each other, right? It's not a competition between in vivo, ex vivo. It's just in vivo AAV is working for certain indications, the right tool for that job, but wouldn't be the right tool for the job we're doing, say, in sickle cell. Nobody's ever proposed using AAV to try to treat sickle cell disease, right? So ex vivo is the right tool to do the job in a certain range of indications, and the data has emerged and demonstrating that. Now, ex vivo comes with some pros and cons and positives and negatives. One positive is we don't have to worry about the immunogenicity issues associated with AAV, so that's a positive. One positive that I've heard sort of interesting from patients, they really, some of the patients I've talked to are really, really glad that we're not inserting a live virus into their bodies. They're really comforted by the idea that you're taking my cells out, whatever you're doing, it's happening in that bag, and what you put back in my body are just cells and no live virus. And so that's comforting as a target product profile. That's a comforting thing for some patients. The other thing that people don't maybe think about as much is ex vivo. Once the cells are outside the body, you have a lot of opportunity to manipulate those cells and to really optimize the conditions before you insert those cells back in. So we have experimented with transduction factors, we've experimented with cell dose. You can do a lot more things outside the body, which you can't do when you're directly injecting the virus in the body. So that's one advantage of the ex vivo approach, and we've gotten tremendous results that we've tried to share. We've gone a step function ahead from where we were with our previous process to our new process, all because of the conditions that we've done outside of the body. So that's one advantage of ex vivo. Ex vivo, though, comes with its own downside. It is more logistically complicated, right? You got to come up with a whole system of taking the cells out, apheresis, shipping it to some centralized transduction facility, doing that, freezing it, then delivering it somewhere else, to get it, reinfused in the patient. It's what Provention had to deal with their product. So that logistically is a lot more complicated than what AAV does, which is like you can just ship vials of that stuff and inject it anywhere in the world. You don't have to deal with transduction facilities and things like that. Another thing that is perhaps more complicated and the downside of the ex vivo approach is in order to get those modified cells to engraft, you tend to have to use some sort of chemotherapeutic approach. You need to do some myeloablation to make space in the marrow for the modified cells to engraft. So, that introduces an element to your target product profile, which means like some patients will be like, oh, I'm glad you're not putting virus in, but I'm not glad that you're ablating me, right? And, with that comes potentially infertility issues and things like that. So that's the downside. Luckily, we're working on diseases that are pretty severe, pretty bad, so people are willing to take that risk for the diseases we're talking about. So, there's pros and cons. I think it's not fair to automatically associate ex vivo with lentiviral vectors. Some people are concerned with the integrating profile of lentiviral vectors. So far, so good from the work that we've seen. It hasn't been an issue. Lentiviral vectors aren't the same as the gamma retroviral vectors that caused the death in the previous study that's famous. But not everybody who's using an ex vivo approach are using lentiviral vectors. Right. We know some people are using electroporation. Right. So there's a lot of different ways to get the therapeutic payload into the cells ex vivo. Lentiviral vector is one way. Electroporation is another way. Other non-viral techniques exist as well. So it's a complicated space once you start putting it together. But once you start wrapping your head around it, it kind of makes sense.
Yeah, Doug. I mean, you've got a broad sense of this. Maybe one more comment from you, and then I'm going to ask the audience if they have questions on any of this.
Nothing jumps out at me now.
Okay. So the goal of kicking this off was just to start with, all right, here's a little bit of discussion on in vivo. Here's a little bit of discussion on an ex vivo approaches. Perhaps throwing out some of the more obvious kinds of challenges and so on. But I want to stop here and see if there are any questions or thoughts. What I really want to have somebody do is stand up and say, that was a bunch of malarkey, you sugar-coated these problems. Let's hear what you have to say about X, Y, and Z. So I'm going to pause for quite a long while here to see if we can get some questions or thoughts from the audience on this. You guys live and breathe a lot of this stuff. Doesn't work, does it? Please, just yell. Oh, you got a microphone?
Branka Mitrovic, Vertex Pharmaceuticals. So in the last 20 years, every time, I guess this is a question for Voyager and Biogen. Every time we do any drug development in the CNS, the first question that we get, the proof of concept in man is just too long. So what are you going to do about it? So that combined with the fact that if you go with the gene therapy in the CNS, while it's definitely directly targeting to what the defect is, you're going to have very limited opportunities to have any kind of biomarker for what your effect is in the brain. So at which point would you be comfortable to decide that you have a proof of concept in men without waiting for the clinical neurological improvement?
I mean, I think from my perspective, I think we're there. I mean, I think, yeah, there are a lot of challenges as it applies to the CNS space. You mentioned one, the lack of biomarkers. But even those are improving, and I think that you would certainly want to build in biomarkers into a program that would help you understand the dose and the other issues as quickly as you can. I think as you think about CNS disorders, you'd probably try to limit the challenges that you have. For example, in certain disorders, you don't need to transduce 80%, 90% of neural cells within the central nervous system, because I don't think that's feasible at this point. But if you could get a bystander effect by transducing a much smaller percentage of neurons, say 30 or 40, or other cells, and getting a bystander benefit, I think those would be kind of the diseases that you would pick first. But in terms of proof of concept in man, I think it's there. I think what we saw today in the talks is tremendously exciting. And I think you're seeing a lot of enthusiasm from larger companies that will progressively get more and more involved. And I think as you get some of these larger companies, hopefully Biogen among them, we bring a lot to the table in terms of disease experience, ability to execute in rare and ultra-rare space. And I think that will drive the field forward as well.
Yeah. Jeff, you want to?
Yeah, I think it's a great question. It's something we've actually been talking a lot about internally at Voyager. I think it is situation specific. I do think it's indication specific. For our Parkinson's disease program, we're fortunate that we have what we feel are really good biomarkers that are going to give us a really clear sense of what the potential of that therapy is. PET imaging, IV L-DOPA challenge test, baseline, and then 6 months out post-treatment. Exogenous L-DOPA dose for a lot of these patients, you should see kind of a reduction in dose. So things could be pointing in the right direction. When we think about the other disorders, it does become a little bit more dicey, and it's an open question. But if you think about the clinical development path as essentially 2 trials where you go, first in man and then some sort of pivotal trial, and what does that first in man look like, and what are you going to learn? Obviously safety's first and foremost, but are you going to have real biomarkers? Are you really going to have proof of concept as we think about it from an efficacy perspective in that first study? The answer may be no. Things that give us comfort is how well the target is defined from a genetics perspective, and I think there are some disorders where that is clear. What we think about specifically as it relates to Friedreich's ataxia and monogenic ALS, which are 2 of our lead programs, there we're targeting the lower motor neurons up and down the spinal cord. What I've been told is there's roughly 120,000 of those. And to think that you're going to go in direct CSF delivery with, call it, 10 to the 12th, 10 to the 13th viral particles, I like those odds that you're going to get, a reasonably high level of transduction of those cells. But it's a tough question, and I don't know if we'll have an answer for every indication.
Right. I think it's one of the reasons, and Doug, you alluded to this earlier, and we'll talk about it maybe before we finish, that some of the initial gene therapy targets, both ex vivo and in vivo, are really around gene targets where there's, I like to say, an unambiguous correlation between the disease and the gene, right? Factor VIII deficiency can be solved with factor VIII. And so first generation gene therapy has been largely focused on areas where there's a monogenic or a single gene relationship between the two. But as the technology evolves, and I think we'll talk about this, where do we go? What kind of potential exists? So great question. Other comments? I can't see.
I have a question. It's Clive Svensson from Cedars-Sinai. I think one of the main challenges when you put a gene into the body and it's expressing is, can you regulate it? Because if it starts doing bad things, then you're in trouble. So if it's RNA knockdown or if it's overexpression of a protein. So I wonder what kind of efforts are being made for doxycycline-induced expression or rapamycin inducible agents, because once you can regulate it with the second pill, it becomes much more of a powerful therapeutic, I think. Particularly, even when you're knocking a gene down, you don't know the bad effects of knocking that gene down.
So gene switches.
The
ability to use, say, small molecules of some sort to turn on and off gene expression.
Correct. How much of an effort amongst the 4 companies is being made for that?
Right. Anybody want to jump in on gene switch? You want to go?
So we agree that I think potentially, particularly for systemic, the regulatory piece is going to be important. But with respect to some of these small molecule regulators, kind of the whole point of doing gene therapy is it's a one-time treatment. And so suddenly you're walking into the world of combination products, potentially an everyday. I mean, if it's just a single on-off switch that somehow you're developing, sure. But if you're taking an ongoing therapy, I mean, potentially economically attractive from your back into the world of daily therapy, but I think that's away from the model that a lot of us are driving towards. And also from a clinical development perspective adds a lot of complication. So I think that's something we think about. I think it's an area that people are focused on, but with respect to those two particular approaches, it doesn't seem there yet to us.
But probably will be more important as you get to subsequent iterations that are not just simple gene replacement, right? Probably not that critical in these simple gene replacement, but when you start doing knockdown or antibodies or who knows what the universe is going to be, 5 or 10 years down the road, your ability to control that transgene with precision is probably going to be increasingly important.
So dose titrate with switches, is that what you're saying? Because I think the question, if I'm interpreting it right, is more on a safety side of things.
That's how I interpreted it, as more as a safety switch off.
Yeah. And my thought is it's a really elegant question. I wonder if it's a question where there's a problem. And at least in the first generation approaches, there's a lot of work that's being done, a lot of toxicology that's being done, and so on. Certainly, if there are problems clinically, commercially, that speak to the need for a kill switch of some sort, then those are the kinds of things that I think will drive it. But at least from my perspective, as things sit right now, the data that exists It's a great question, but I don't know that there's a problem yet, or at least I haven't seen it as a problem. The other thing, if you then turn it back to existing vectors, not lenti but AAV, not that everything, but loading capacity, it's something we didn't talk about, right? But the basic capacity of AAV as it exists is very limited, and the ability to put in your transgene plus an effective promoter and anything else you want to put in there to package, there's not a whole lot of room for a lot more in terms of small molecule switches and so on. That's not a good answer if there's a need, right? But in terms of the existing systems that are broadly being used, and AAV, not limited, I agree, only AAV, but broadly being used, it's one of the realities that we would face in order to do that. But I think it's a great question and one that deserves discussion. I'm not want to dominate the point.
But Clive, a question back to you or anybody else in the audience. On the stem cell side of things, what about kill switches and ways in which you can immediately get rid of a rogue stem cell population? Are people doing that? Have regulators opined on that?
Well, it's been very complicated, right? Because the complication of putting the kill switch in is more regulatory hurdles to go through. I think my concern is more if you put a protein in and overexpress it, as Voyager's doing, or you knock something down, as you're doing with the SOD1 trial, you don't know the long-term effects. So it would be very nice to be able to say, or you might want to pulse the delivery, for instance. Give it 2-3 times a week if you're delivering a therapeutic protein using a gene therapy approach ex vivo. So it's just a safety factor, but I completely hear what you say. If there's no safety concerns in your preclinical models, why go to all the effort? The only thing I would say is in humans, it's not just a 2-year or 6-month preclinical trial. It's actually for life, right?
Right.
So once that gene's expressed, patients could, after 5 years, have an effect of knocking it down or overexpressing it. And that's the point at which you want to say, gee, I wish I could turn that off. And I just think that's something that the field needs to accept. When you put these cells or anything into a patient, it's not just for your 2 months or 2 years for your study trial, it's for life.
Yeah, the point's very well taken. I mean, I think in the French trials, it was a couple of years before the old retroviruses began to show, adverse events in leukemia and insertional mutagenesis. So the point's well taken, and I think it's an important one for the field. So thank you for raising it. I'll put it under the heading of we sugar-coated that one, and I'm glad you brought it up. So other points on this issue of things that you're curious about or want to get the perspective of the panel on in terms of either in vivo or ex-modified cell therapies. Please.
I'm Owen from Discovery Investing as a perspective from investors. I guess, when do you think is the right time for us to enter the gene therapy field?
When is the time for investors to enter the gene therapy field?
Yes, sir.
What time is it?
2013.
I love it. So you want an honest, you want a more direct, less glib answer. I think, I mean, it's your money, right? My point of view is it depends on your risk tolerance, depends on how much data you want to see, and anything else. But I think we're, the point now where, we should be evaluated just like any other drug development company. Do we have data around a product that's sufficiently de-risked that give you some perspective on value creation and so on? So anybody else want to ask about when they want to invest?
Well, I think even over the next 12 months, there will be substantial de-risking of this field. We saw in the presentations today that there are some very compelling proofs of biology and proofs of concept that are going to read out quite soon. Now, there will be longer-term challenges. We won't know if the durability of the effect and all of the things that we spoke about, there still will be challenges, and we may kind of have fluctuations in the field where we may have some irrational exuberance up front, and then something's going to happen. We've seen that with this field before, where you may then take a step back and say there are some challenges that we had not anticipated. But in terms of an inflection point for valuation of the field, you're going to get that really soon. Whether it stays up that high for a long time depends on things that we don't know yet.
Yeah, I agree. I mean, I think the things that are going on here are incredibly exciting. There's a lot of work going on, but there's going to be a lot more bumps in the road, period, paragraph. And it depends on your risk tolerance.
Yeah. Do you think we're still at a place where a setback in one area flows into all the areas of gene therapy? Because my experience, question to be answered, because as you point out, I think there's key data coming in the next 12 months, is investors seem to me to be getting much more sophisticated. And so potentially, depending on what happens, that they're, that the field is being judged more maybe strategy by strategy.
I think that's right. I think there is more sophistication now, and it would not cast a stain on the entirety of the field, right? It would cause a kind of a nuanced reaction, which says, well, okay, we learned something about that, and we maybe want to shut down one particular area, but I think it's unlikely that something would happen that would just shut down the field entirely.
Yeah. I was talking to Rennie Benjamin about this. You posed the same question yesterday to the finance panel. I was like, do you think we're at that point? And it's interesting because I think it's a technology by technology and indication by indication thing. Take the CAR T space, right? The MSK had 2 deaths in their study. And, that's lentiviral vector, gene therapy, and though most people don't think of it that way, there were 2 deaths. They had to put their program at clinical hold, and they got past it. There was no panic, right, that happened as a result. And I think, frankly, because a lot of people don't associate CAR T with like lentiviral vector gene therapy, they're just CAR T technology. They don't even realize it. They don't think of it as like this is an HIV virus which we put inside T cells. So in that case, it proved not to be sort of something that was like a watershed moment. But on the other hand, maybe in heaven forbid, if in our sickle cell disease program somebody developed leukemia, that could be like a big deal. Right, and we don't think that'll happen, and we hope it doesn't happen. But so I think, so I think it's really we're still not sophisticated enough. I think, and not all investors are sophisticated enough that I think there is a risk, depending on the event, depending on the indication, and depending on the investor or the class of investors, you may have sort of panic, run to the hills, and in other situations, I think the field would be able to absorb it.
Where are you on that, Ed?
I think it's one of these questions where we don't have to speculate. It'll happen. It'll happen. I mean, something serious is going to happen at some point, given all the work that's going on, all the things are going to happen, and we'll see it. We'll know.
I think what I'm struck by is, and this goes back, 15, 20 years ago, kind of the first go-around.
Yeah.
We talked about on the prep call what's different today, and I agree the level of sophistication is dramatically different on the investor side. And I'm reminded of what my mother said when I was growing up: just because Johnny did X, doesn't mean you have to do X. But if you go out and you talk to whether it be venture investors, whether it be buy-side investors that are focused on crossovers, obviously there's been tremendous interest from a public market standpoint, whether it be strategic collaborations with larger biopharma companies. I mean, there is a significant amount of investment flowing into this space. So, to Edward's initial point, the time is absolutely now.
Yeah, I think it is. All right, thank you. Was there another question? Good. Yeah, please. Never heard of it. Well, I mean, I'm happy to talk about it in great length, but does anybody else want to jump in on that?
I think that was
directed
to you.
That was for you. So for those of you who don't know, the field of genome editing has a couple of approaches. Approach that Sangamo is involved with are zinc finger nucleases. There's another platform of tal nucleases, and then more recent efforts in CRISPRs. I think that if you take the opportunities and challenges thing, the way we were talking about in vivo, ex vivo, the opportunities are to do permanent genome editing with these platforms, and the kinds of potential clinical outcomes that can drive, I think, are motivating and interesting and potentially very important. The challenges to make these platforms work are significant. If you distill them down to the greatest, most important ones, particularly as it relates to human therapeutics, I think it comes down to the ability to build these nucleases that are very, very, very singularly specific to their targets. And so you have an issue of specificity versus off-target effects. And And right now, there's a lot of data around the most mature platform, which is the zinc finger platform. There's less data around the others. But the hope is that these technology platforms can do genome editing in a way that can drive permanent genetic changes, literally human genetic engineering, to endogenous genes that can cure diseases. So it's an exciting, hot space of which we're delighted, Sangamo, delighted to have the visibility that CRISPRs have created. Was that sugar-coated? Who knows? Good. Well, I wish I'd cut off the questions earlier. All right. So that was good discussion. I appreciate that. So let's turn to kind of the more business side of things and see what if this is of interest to you all. But in our prep call, and it turns out that Hans in a previous life was a patent lawyer and came up through that side of thing. And I know in my life I've spent a lot of time thinking about not just the technology and what we're doing, but the intellectual property that surrounds it. And so we had a good discussion on our pre-call about is that relevant or not? And I've seen things where I get wrapped around a spool about how important it is that we have patents, we have freedom to operate, and so on and so forth, and not, and so on. So would you mind talking a little bit about, kind of your perspective and maybe industry perspective? And then, Doug, I'd like to get your thoughts about when you look at technologies, where does IP fit in and so on?
So thanks for outing me as a
former
patent lawyer. So we talked about a couple topics, and I think two of them in particular are good for us to discuss, and then two I'm just going to kind of quickly tie off. We talked about is important, so I think that one is relatively uncontroversial. And then does it drive product decisions? How does it impact your partnering decisions? And then there was a question of what about Myriad?
Yeah, the Myriad decision.
Look, I think with respect to is it important, along with clinical data and clinical progress, I think IP is one of the foundations of the industry. I think our philosophy, and I think many people's, is that great ideas deserve to be protected, as well as the investments that we're all making to, develop those inventions. That's where I'm at on that. To the extent that there is a countervailing view and that we should be in a public source zone, I'd solicit that from the panel, but I'd be a bit surprised.
Not for me.
So, what about Myriad? I'm just going to tie that off as well, which is, the recent case on compositions that read on naturally occurring products being patent ineligible. That made some waves, and this is an area where I think the law feels a bit unstable to me and is evolving. I think in the recombinant space, where you're doing something that's non-naturally occurring, a lot of the things that we were talking about today, I think it's not so much of an issue, but I think in the area of cell therapies, personalized medicine, diagnostics, some of the autologous methods for separation, you have to pay a lot of attention to it, and I think it's going to evolve, and hopefully we'll get more clarity. The Patent Office has developed a 14-point test in an internal memo that is now widely circulated that I think provides some sense of a roadmap, but frankly I think is a challenge and adds complexity when people are looking at one-on-one topics. So with those preliminary matters addressed, I think it'd be interesting to hear the panel and solicit questions on how IP impacts product decisions and partnering decisions. I think on product decisions there's a diversity of opinions and philosophies on how people approach IP. For us, it's a consideration, but I'd say it's a secondary consideration. So, I think we feel that if there's good biology and understanding for a target, there's a strong unmet need, we're going to want to find a way to make that product if we think it's central to our strategy and is going to help patients.
So if there's something out there and you think, 94% that's going to cover us, but it's you're not going to necessarily stop.
So good question. I think we're going to scrutinize the IP carefully.
Yeah.
Right. I think there's a balance between respecting bona fide IP rights, and I'd say that's the other important part of our philosophy. But at the same time, I think there's a lot of ways to be creative about developing the nice thing, particularly about AAV, is because it's a mature field, there isn't a Billy patent, there isn't a dominating, overarching umbrella patent that sort of everybody has to go through. And so I think there's a lot of ability for people to be creative and develop workarounds or have unique insight that allows them to take another approach. And so, at least from our approach, that's something we look at carefully because, of course, you need to protect your investment. But at the same time, the biology and the clinical need is our driver. And
so they come to you guys, Doug, and they've got a product that makes tons of sense. They can make it. It works. They've characterized it. They've got data. But there's a hole somewhere in the IP. How much does that bother you?
I mean, it does to some degree. It does feel a little bit uncertain and fluid at this point. I mean, and there are several considerations, right? Some of the core foundational IP is kind of 2023, 2026. And that's not a whole lot of time, especially in...
I will definitely be dead by then. It's not even close.
And some of the other IP is kind of annoying to others, but they can make workarounds on it. You're just kind of throwing stuff out the back of your truck to slow people down and get them to swerve a little bit. but they can. And you try to throw a couple of them out, and it slows them down a little bit more, but ultimately there will be a workaround. You will get some regulatory exclusivity, which in many of the disorders that we're into. And so that certainly makes it more attractive. So, it feels a little uncertain, but it does feel like the biology is really driving this to a large degree, and that, we certainly feel like we can get our investments back, that we can go into diseases, that we not only have freedom to operate, but we can get a return on the investment.
Yeah. I mean, you guys are both, parental sort of things, Third Rock and that Boston group and so on. I mean, the sense, I'm sitting out in California, right? But the sense of what I have with you guys is, if it's a good idea, you guys are going to go after it and maybe worry about picking up the IP pieces after you've determined whether it works or not and so on. Is that a fair way to think about things?
The way we thought about Voyager when we were creating it inside of Third Rock was there was certainly an appetite and a desire to have what we call kind of foundational IP.
Yeah.
If that proves out to be, critical IP from a product program perspective, is still a little bit to be determined. We brought in a whole host of rights to a number of different AAV variants, and if one of those variants ends up being nominated as a lead product candidate, great. Not 100% clear yet. I think, we spend a lot of time thinking and talking about IP internally. It's complicated, and it's a little bit of a to-be-written story. And I think if I had to delineate between two, and we're talking about a multifactorial product, right? You have the capsid, you have kind of the promoter element, the transgene. I think
manufacturing.
Delivery. So it really cuts across all those. I would say where we spend most of our time focused on is obviously the capsid IP.
Yeah.
And there's the historical IP covering wild type serotypes or maybe some newly engineered variants. Our general view, though, is from an evolution standpoint in terms of the field, just scratch the surface. Hans presented a lot of the work they're doing on directed evolution, very large libraries with novel variants. We think, as a field, you're going to see an absolute explosion of these new novel variants that may or may not be covered by prior IP.
Yeah.
And
that's where Myriad also
view,
folds
it's going to become
back
more
into this.
commoditized.
Yeah.
I'd say we take IP pretty seriously, actually.
I know you guys do, yeah.
And we sort of made sure, first of all, IP in this space is a lot more complicated, I think, than in other spaces because of everything we talked about, right? There are things associated with the virus manufacturer, the transduction, delivery, everything. So it's a pretty complicated landscape. One of the first things that happened when Third Rock recapitalized the former Genetics Pharmaceuticals and created Bluebird Bio was a year-long effort to sort of clean the space, make sure we had freedom to operate, and had the platform on which then we could pile on other indications with freedom. So we know we have freedom to operate in the things we're working on. When we started looking opportunistically at other tools, which eventually led to the Progenin acquisition, we took a very serious look at different indications, what's the right tool for the job, and then how many of those tools were even available, right? And a lot of the AAV tools are taken. CRISPR was getting wrapped up under Editas. And then in Progenin, we saw a portfolio of very interesting gene editing constructs, broad IP portfolio, freedom to operate.
Right.
So I think Bluebird Bio has been conservative and very responsible, and so make sure that we have a very clean slate, but a broad slate on which we can pile on a lot of indications.
Yeah, good.
But Hans, so your take on the AAV variants, the directed evolution, and the protectability of them down the road?
Oh, sorry. So don't get me wrong. I mean, I think I was really sort of talking about sort of how we sequence our decision making, but I agree with Jeff, which is that the capsids are really equivalent to your molecular entity in a pharma setting, and is really going to drive your kind of sort of core IP. So yeah, I mean, I think they're protectable, but I also kind of agree with Jeff that the landscape of sort of is this sort of a multi-solution landscape where lots of capsids can do the same thing, or really there's some unique spaces within that diversity that I think that's to be understood.
Yeah. I think the read-through there from a commercial perspective is first to market is going to have a huge.
Well, that was really the flip side of all this, right? Is that relevant? And so let's use that maybe as a transition to talk about beginning to move towards commercialization and so on. And there's been a lot of discussion here, good discussion on reimbursement, pricing, value creation, and so on. And Faraz actually chairs the reimbursement committee for ARM and has done a lot of work in this space. But maybe could you start, if you wouldn't mind, on this issue of kind of value of being first to market and where or why that's a different or not in the cell and gene therapy space?
Yeah, I mean, it's a good question, and it's something worth thinking about for anybody who's sort of entering a space. And if there's 2 or 3 people ahead of you, should you even be going into it, right? So,
well, particularly in orphan situations.
So it's actually that. So the value of being first to market is higher than ever.
Right.
Orphan diseases are already small by definition, and unlike the first iteration, the enzyme replacement therapies, well, if you come up with a follow-on enzyme replacement therapy, there's an installed base that you could penetrate into. But when you're talking about curative therapies, once you start curing these patients with orphan diseases, then you're just shrinking that installed base. There is no installed base, right? So if you're second or third to market, and there are a bunch of people who have already been sort of effectively or functionally cured by the disease, you really. So there's a tremendous value being first to market, specifically for curative therapies, specifically for orphan indications, and particularly because in orphan diseases you can get 7 years of exclusivity,
right,
in the U.S. and 10 years in Europe. So you got a long time. Where if you've got a good corrective gene therapy and you could sort of block others from coming in because of the orphan legislation, during that entire period, you might just clean up the market, right? I'm just, nothing is ever that clean, but I think the value of being first to market for orphan diseases, for curative therapies is particularly high. Having said that, it does come down to your target product profile. I mean, I think the Hep C has provided a great example of why. Incivek came out, right? Incivek, Vertex launched Incivek. It was a great launch. It had a profile. The profile was sufficient to treat certain number of patients, but a lot of patients were were still left over, and some patients waiting in the wings for then when Sovaldi came in. And now Sovaldi's making a tremendous penetration, but there'll still be patients left over, even though it's a curative product. There's a next generation product coming right behind it. So, I mean, that's an example where curative therapies have come in one after another, but they come in with different profiles. And to make that tangible in here, let's just say that we come up with maybe we're first to market for beta thalassemia or sickle cell disease, but let's say our approach is highly myeloablative, and there's a good pool of patients or physicians who say, I don't know if I want to go through that. And then somebody comes up with a follow-on product that's non-myeloablative. Everybody would be interested in a product like that. Some people may say, I'm going to wait for that, next generation product. I can afford to wait another 3, 4, 5 years for that next generation product, and then I'm going to jump in and get treated. So on one hand, the value is very high for being first to market, and on the other hand, I don't think that means that if you're second or third to market, if you've got a superior product, I think there may still be a market for you and a healthy market for you to go after.
Agreed. Doug, when you guys think about the more rare and orphan diseases, how do you guys think about this issue of
get there first?
And just marry that with the earlier discussion of barriers to entry, freedom to operate, and then in terms of first to market product differentiation. I mean, it's sort of a matrix of thoughts, but I wonder how you guys think about that.
Yeah, I mean, the barriers to entry, I mean, so it depends how good you are within a rare disease space, because you may be offset in this ultra-rare space by just having an awful tough time knowing the natural history of that disorder, recruiting patients, getting centers with expertise, or getting clinical endpoints that are approvable. So that's the downside, right? But yet you have very good protection if you're in that space and you get to market first. You know, somebody could break your exclusivity, but it's hard, right? Superior efficacy is going to require probably head-to-head. Safety you could get in some circumstances if you have a, you know, myeloablative approach or something like that. And then tolerability, if it's a really substantial burden to the patient, would be another way to break that exclusivity. But it's a really high bar. Good. So you strike that balance of, you know, huge unmet need, very severe disorders, first to market, as long as you can recruit and have valid endpoints and work through the natural history as a really good entry.
Let me just pause for a second. So we've only have about 10 minutes left for the panel. I want to wrap up on a more forward-looking kind of thing. But are there questions that you all have about in the macro of commercialization, intellectual property, pricing, reimbursement, anything that maybe came up in other sessions, that, or just any questions you want to bring up on those sort of topics? Please.
AG orphan for MS, for beta serum, and then Biogen came with almost identical drug, and orphan status was struck down. For the good of the patient, better drug, better profile. So unless we're talking about extra, ultra orphan diseases, if you have a better drug, it doesn't seem to stop the development before. I guess MS is much better, much bigger disease than some diseases you guys are discussing, but after they had orphan, there were 10 drugs that came after. Well, 8, basically. So orphan, it really depends a lot what's good for the patient and how the company behaves and what the drugs are coming after. So
yeah, it's a good point. I mean, orphan isn't the end-all, be-all from
right.
I
mean, I don't think it stops anybody.
Yeah. But you know, the example that we use most often internally when we talk about this issue is think about it from a practical perspective. ALS is a great example, right? If you have ALS with the SOD1 mutation, you know, the unfortunate reality is those patients are going to be dead in a couple of years, maybe even less. And if you're first to show a meaningful benefit from a clinical development standpoint, you know, translate into kind of the first approval, how are you even going to enroll the second trial for the second product? I mean, if you're sitting there and you were just diagnosed as a patient, would you want to be taking the experimental therapy, or would you want to take the real thing that was already approved? So I think it is kind of multimodal in terms of the protection. But I think they all lead to the same place, which is, you know, getting there first is going to be pretty important.
Right. So just in terms of time, let me turn to sort of last topic. What I asked, or what we talked about on our pre-call, was to take a crystal ball out and step back a little bit and say, okay, the first generation products we've talked a little bit about, many of them are protein replacement or monogenic or validated disease targets. As the technology evolves, and we've talked about some of the things that are going on there, what do we see 3, 5, 10 years down the line that the areas of gene therapy, genome editing, ex-vivo cell modification can begin to address? And Doug, would you mind kicking this off? I mean, you're sitting at Biogen with a strategy hat, right? And thinking about these things. So,
it's fun because you don't know what the universe could be of this technology and what payloads really you can think about. But I mean, just a couple that people have thought about. So antibody delivery, right? So that would be right in our wheelhouse. Biogen is a deliverer of biologics, especially to the central nervous system, where we have challenges getting those biologics in meaningful concentrations into the central nervous system. So asking an AAV or a lenti as a gene therapy delivery vehicle to give, you know, antibodies or biologics against amyloid or other relevant things would be would be a game changer, I think, for us. And then I think the other one that people are thinking about, we're certainly not unique in this space, is RNA modulation, RNAi, microRNAs, ways to modulate these very complicated processes, to knock down genes, to alter their regulation, alter their epigenetics. But I do think, as we talked about before, that once you get into that universe, you probably are going to have controls so that you can upregulate and downregulate your payload, because we don't know 3, 4, 5 years down the road if you're knocking down something 70 or 80%, whether or not there's going to be consequences to that. And so I think another layer of sophistication is going to have to be built in, assuming that the viral vectors allow it.
Capacity-wise, loading capacity-wise.
Loading capacity-wise may be challenging.
Yeah, I think that's absolutely true. And Hans, when you think about some of the molecular evolution you guys are doing, it's not just capsid proteins. You're looking at other elements of the vectors as well, right? Do you see going forward the ability to expand packaging capacity in AAV, or is that an area of focus? I mean, it's certainly one of the areas of limitation right now.
It is. If you think about ASGCT this last year, there was a lot of people having interesting ideas about combining payload out of different vectors, so that may be one way that it goes. That's not, for the moment, for us at least, there's so many things that we can do with that. It's sort of more than we could ever do, that it hasn't been really a functional limitation for our
I agree with that. I mean, I think one of the other opportunities we've been thinking about and working in is more synthetic vectors, right? Things that have the kind of ability to have various tissue tropisms with various loading capacities, with the potential to target, and then if and this is all way off, right? I'm not suggesting we're gonna. This thing's not webcast, right?
I think the next wave is going to come in because of 3 different factors. So one is, right now we started with, for simplistic reasons, right? We started with a monogenic, simple, one gene replacement. It's a simple, clear story. So the one obvious evolution is you go, once you've gotten your proof of concept clinically in these monogenics, then you go to more complex diseases, right? So that's one form of evolution. Another form of evolution will be because of the convergence of technologies. You can package more things into this thing. With lentiviral vectors, there's a bigger carrying capacity. And homing endonucleases, which we acquired, they're small constructs. You could actually load up several homing endonucleases to deliver different genes, to do different functions, all in one lentiviral vector. Yep. So that's a technological leap that will allow the next generation of indications. The third one will be more business model driven. And what I mean by that is they're currently, let's remember, there's 6,000 rare diseases. Most of us end up talking about the same, say, 20, right? And why a lot of people don't go after some of those other ones is that they're so rare. And then you go, God, the price tag I'm going to have to come up with to sort of justify going after those ultra rare ones is going to be, the sticker shock's too high. But if we can come up with some of these novel pricing and reimbursement mechanisms, say, well, there's a way to address that and create the right incentives, create the right pricing and reimbursement models, then suddenly there's going to be a whole pool of diseases that were previously considered commercially unviable or that we might be able to actually go after. But it is going to require reinsurance schemes or pay for performance annuity or something to smooth over the sticker shock of ultra high prices for a very, very rare condition. But that's very deserving because it's a high unmet medical need and the technology is there.
And I'll just pile on a little bit on the business model driven changes that can come technically, and that speaks to the whole issue of autologous cell therapies and evolving those towards allogeneic cell therapies in a way that is, why don't you do that now? Well, because you have immunological issues. Can you create a universal donor with a universal cassette into which one can then put in whatever one of your 5,000 favorite gene replacements are? That can be done in a centralized manufacturing facility in the absence. And so the ability to do, from either a business or clinically driven rationale, to change the business model to change the technical model of a autologous-based business to an allogeneic-based business, I think is something that I think is very worth considering and potentially doable in this 3 to 5 year time frame. We will see. Good. We have just a few minutes. Any last questions or comments? Well, in that case, I'd like to thank the panelists. If you'll join me in thanking the panelists, I thought it was a good session.